Right, welcome everybody. It's my pleasure today to introduce our speaker, Marshall Brennan. Marshall was born in New York and studied at Northeastern University and Harvard, where he also carried out research in the laboratory of Professor Richter. As an undergraduate, Marshall also spent several months working for pharmaceutical industries, including Novartis and RXI. He obtained his bachelor in 2011, and then he joined the program here at the University of Illinois, where he's now working with Professor Falter. And he's working on the synthesis of first-row transition metal complexes for two-electron processes. Today, Marshall will be giving a talk entitled FeMo Cofactor Characterizing Nature's Carbon.
All right, well, thank you very much, Andrea. It's a pleasure to be here to discuss what I think is a really fascinating story of characterizing the iron molybdenum cofactor of nitrogenase. And so the FeMo cofactor, or FeMo-Co as I'll be referring to it over the course of this seminar, is arguably one of the most complex biological metal centers known. And it consists of two cubane type metal sulfide clusters bridged by 3 sulfides and this X ligand that will be the concern of today's seminar. And so this X ligand has been the source of a lot of controversy because early on it was very difficult to characterize. And as the name of my title of the talk gives away, it's been recently deduced to be a carbide. And I'd like to take you through the experimental studies that determine this. So FeMo-Co is the active center of nitrogenase, which is shown here and is a C2 symmetric homodimeric enzyme that is found responsible for bacterial nitrogen fixation in the Clostridium and Azobacter species. Nitrogenase has really fascinated scientists for about the past 50 years since it was identified, primarily for its role in introducing nitrogen into biological systems. So shown here are the amino acids and nucleic acids, which are arguably the most common building blocks you'll find nitrogen in. And to take the amino acids as an example, they build the key structural elements in proteins, and here myoglobin is but an example of how these peptide bonds form these large architectures that perform mechanical function in cells. And in addition, they perform Lewis basic functions in the large natural products such as the alkaloids, a member of which is voacamine, shown here, and is a potent cytotoxic agent found in marine organisms as a defense mechanism. So amino acids themselves, if we track them back, ultimately come from the biosynthetic pathways in plants arising from ammonia. And ammonia is what we call the lowest nutritionally available source of nitrogen. That is, it is the simplest molecule that can be taken up by plants, and the plants are subsequently using this to perform biological building blocks that are introduced to the food chain through the consumption by herbivores and subsequently carnivores. However, in a stroke of irony, plants cannot produce this, nor can they sequester it from the environment, and instead they need to get it from bacteria that contain nitrogenase. They perform what we call nitrogen fixation, which is merely the reduction of dinitrogen into 2 equivalents of ammonia. And this process breaks what is one of the strongest known triple bonds between nitrogen and nitrogen. And what I won't be able to get into in the course of this seminar, but is actually really important to know about this enzyme, is that it actually continues to cycle through and is responsible for the nitrification of ammonia and the subsequent oxidation and denitrification to complete what we call the nitrogen cycle. And this is how dinitrogen is both sequestered and used as a building block and subsequently restored to the atmosphere, both breaking and reforming the strongest triple bond that you can expect to find in a natural system. So this can be compared to how we as scientists do this same process in the laboratory, and using the Haber-Bosch process introduced by Fritz Haber at BASF in 1910, we can take dinitrogen, convert it to ammonia using temperatures in excess of 400 degrees Celsius and 300 atmospheres of dihydrogen. Now, despite this being a relatively energy-intensive process, it has had an incredible impact on human development over the past 100 years, and has been directly attributed to the tripling of the population between 1900 and 2000. And shown here is a line indicating about when the Haber-Bosch process was first put into motion. And you see a meteoric rise in population over the course of the next century. And it's important to note that the reason why this fascinates us is not because we want more people on the planet, but because the huge energy demands of this process are currently sequestering an enormous amount of fuel. And if we could begin to mimic how nature does this and instead produce ammonia in a more energy efficient process, it would free up fuel reserves for other purposes. And so with that in mind, today's talk will first introduce you to nitrogenase in terms of its structure. I'll then tell you how its components were identified with respect to their function, and that'll tell us about FeMo-Co. And through discussing its early crystallographic studies, this will tell about the ambiguity regarding its structure and the controversy surrounding whether that X ligand I showed you earlier was an oxide, a nitride, or a carbide. And I'll end the talk by talking about some very recent work into the biosynthetic origins of this ligand and the implications it has for future research into its mechanism and a more energy efficient Haber-Bosch process. So it's worth mentioning at this moment that there are actually 3 known nitrogenase enzymes. The one that I'm going to be spending the entirety of the seminar on is known as MONASE, or the molybdenum nitrogenase. And if you starve the bacteria that produce this of molybdenum, they'll then begin incorporating vanadium into that same spot capping the structure. And it retains its nitrogen-fixing properties, but you see increased promiscuity toward the reduction of protons into dihydrogen. If you further starve it of vanadium, you'll incorporate iron instead. And both of these enzymes are incredibly unstable, and as a result, they're not very well characterized. But these structures are proposed to be nearly identical to that of FeMo-Co on the basis of spectroscopic studies. So nitrogenase here is shown as one of its 2 monomers. And up top, we have the iron protein. The iron protein provides the electrons that are subsequently used in the reduction process at the FeMo cofactor. Between this is the physical and electronic link between the iron protein and the FeMo cofactor, the P cluster, which oxidizes in response to the binding and subsequent reduction of dinitrogen at the FeMo cofactor active site. And so these parts were originally identified by Shaw and co-workers in 1977, and what they did is they broke the enzyme down into its constituent parts using a mixture of sodium chloride and citric acid, and then separated them chromatographically by gel electrophoresis, and used an assay using the reduction of acetylene to ethylene to identify the redox active portion of the enzyme. And they found that the fractions containing a compound that by elemental analysis and mass spectrometry matched 7 irons to 1 molybdenum. Those fractions were the sole components that could reduce acetylene, which is, of course, a secondary reaction of nitrogenase. And so they termed this the FeMo cofactor, and then showed that if they reconstituted the enzyme with the FeMo cofactor from one species, with the iron protein and P cluster from another, the reconstituted enzyme actually retains the almost identical activity profile regardless of what combination you use. And so the thing to keep in mind over the course of this talk is that it's thought that there's effectively one nitrogenase that was evolved and then evolved into several different species of bacteria. And so over the course of this, there's generally been the favorite bacterium in these studies, Azobacter vinlandi, but nearly all of the bacteria that produce nitrogenase are expected to produce very similar, if not identical, enzymes. So the first crystallographic study of this enzyme came from Doug Rees in 1992. And it was a 2.7 angstrom resolution structure. And if we look closely, we can see the iron protein, the P cluster, and most importantly to me, the FeMo cofactor. And we see our 2 cubane structures. The molybdenum here is depicted up top. And the 3 bridging sulfides. But most importantly, what he noted was that in the center of this cluster was a bridging ligand of light electron density. And in 1992, he formulated this as either nitrogen, oxygen, or sulfur. And the next year, he produced a higher resolution crystal of 2.2 angstroms that was not able to refine this assignment any better. But in both cases, the iron was thought to have a trigonal coordination sphere. And so Rees favored sulfur. But almost 10 years later, he produced a 1.16 angstrom structure, which was for a long time the best crystal structure available, which showed that it could not be a sulfur on the basis of the observed electron density, which refined the iron coordination sphere to tetrahedral, and instead included carbon as a possibility. And the question was posed of, is this a carbon, nitrogen, or oxygen? And for those of you who are wondering why this was a concern, is that a common problem in Fourier analysis is that you observe these resolution-dependent ripples in electron density around heavy metal nuclei. And so it can be described by this formula, but pictographically shown, we see that 6 iron nuclei in direct contact with this ligand provide a ripple that additively spans actually a very wide range at this resolution. And so on this picture over here, we see the positive electron density for the iron atoms. This inner sphere represents the positive electron density from the X ligand. And this region in between is effectively completely ambiguous, because it appears that there's not only no electron density, but negative density. And those 6 iron atoms are completely obfuscating the characteristic electron density from this active site. And so here is where we find our controversy, because this bridging X ligand has some implications that could potentially inform the mechanism of this enzyme. Because at this particular moment, even today, the mechanism of this enzyme is unknown. And it was very appealing to think that if this X-type ligand was an oxide, one could postulate that the inclusion and extrusion of water could provide a thermodynamic driving force for this reaction. If it were a nitride, one could expect that perhaps the nitride arose as a byproduct of nitrogen fixation. And of course, the possibility of a carbide was not really well understood, and arguably is still not very well understood. And so it didn't receive quite as much attention over the course of this inquiry. And I'd like to go over, at first, how they came to the initial conclusions that it was a nitride. And what you'll see is that oxygen was ruled out very early on as a possibility as a result of Dantz's work, which I'll get to very soon. And that in the first few years after this publication of this crystal structure, the work went almost entirely toward identifying a nitride ligand. And it wasn't really until about 2006 that the possibility of it being a carbide was really taken seriously, and not until last year that spectroscopic evidence really provided any data to conclude that it was in fact a carbide. And so in the ensuing few slides, I'll be discussing the evidence for a nitride-based ligand. So the year after the crystal structure was published by Rees and coworkers, Ian Dantz at the University of New South Wales in Australia did a DFT study that, where he studied the geometry-optimized structures of a putative FeMo-Co model that included both a nitride and an oxide bridging ligand. And what he found was that a structure that had a bridging nitride retained the symmetric properties of the enzyme relatively well, and the calculated spin state of 3/2 matched the experimentally observed spin state of the enzyme as well. And so the data was relatively well in alignment, but of course an oxide was found to greatly distort the distances in the experimentally observed structure of the iron atoms. And of course, the oxide was found to fall off-center and break the observed symmetry in the crystal structure. And so on the basis of this data, Dantz ruled out the oxide as a potential X ligand pretty much immediately. And this was one of the first really important papers in discussing what could be done to identify this ligand. And perhaps it was the end of this work where he studied a potential mechanism for the inclusion of this nitride ligand that inspired a lot of work into discovering a nitride ligand in the center of this active site, where he found that were one to form this bridge dinitrogen structure, reduction of this one of these nitrogens to an ammonia and the other to the nitride ligand was found to be highly exothermic on the basis of DFT calculations. And so this, of course, was in line with the idea that maybe this could shed some light on the very ambiguous mechanism. But of course, it was based entirely on the assumption that the bridging ligand was nitride in the first place. And so it should be mentioned that he didn't actually consider a carbide at all. And so in the very first paragraph of this paper, excluded on the basis of it being unreasonable to expect a carbide, a C4- ligand, to exist in a biological system. So the same year, Norskov and co-workers did a little bit more thorough of an analysis where they made the same nitride and oxide model structures, but also made two structures that could be attributed to having a bridging carbon ligand. One of them was a methylene and the other was a carbide. And what I've done here is I've taken his data from his computational models and subtracted the experimentally observed crystal structure internuclear distances from those models to show you the deviation that each model structure has from the actual enzyme. And the first thing that jumps out is that if we look at nearly every metric, the oxide falls out as the highest degree of error, and down here is the average sum of those errors. And so this was in direct correlation with Dance's observation that an oxide ligand was, for all intents and purposes, unreasonable on the basis of structural data. Now looking at the next highest bar in this rightmost column, we see that the nitride is actually the second least congruent structure with the actually observed enzyme. And Norskov doesn't really draw a lot of attention to this, especially that the carbide is the highest correlation with the experimentally observed structure. And I think the reason behind this is that he also calculated the expected binding energy for the X-type ligand into a putative unligated FeMo cofactor complex. And so in silico, he reacted a iron molybdenum cofactor without an X-type ligand, which I've just summarized here as Fe7 molybdenum, with a protonated X-type ligand, and then produce a ligated bridged FeMo cofactor with those protons lying on the iron atoms, postulating that while the magnitude of these bars cannot be taken with respect to the biological system, the direction of their enthalpies is relevant, with the nitride and the oxide ligand finding an exothermic binding profile. But the carbide and the methylene structures were both highly endothermic in their formation under these conditions. And so despite having the appropriate spin state to match the experimentally observed structure, he ruled out the carbon bridging ligand in favor of a nitride. So further density functional studies came from Noodleman and co-workers, where they modeled the bound oxide, nitride, and carbide ligand and measured their expected redox profile, and found that the oxide and the carbide were not in very good correlation with the experimentally observed redox potential, but a bound nitride was actually reasonably close. And so on the basis of computational data, it seemed like a nitride was a reasonable conclusion to draw. But in terms of spectroscopic data, the early work was really difficult to approach because there are so many spin-active nuclei in the presence of the originating X ligand that it's difficult to observe by NMR. And so instead, a popular technique came in the form of Endor spectroscopy. And for those who are not familiar with the technique, electron nuclear double resonance, Endor, answers the question of what nuclei are coupling with my paramagnetic center. And to give a brief overview of how this technique generally works, we get an NMR resonance from an NMR active nucleus such as a carbon-13, and then we saturate its EBR signal. And then when saturated, we get a spin coupling event that splits the NMR resonance, and this coupling is measured out in the EPR spectrum as an Endor line. And so this tells us, actually at reasonably high resolution, which types of NMR active nuclei are binding our metal centers. And so Hoffman and co-workers did just this, where they performed an Endor analysis on Azobacter vinlandi species that were grown in N14 dinitrogen, isolated the enzyme, and found that there were 4 nitrogen nuclei that could be attributed to a potential X-type ligand. And in response to Dance's postulation that this is involved in the binding and reduction of dinitrogen, then incubated that enzyme with N15 labeled dinitrogen, found no enrichment of those signals, and concluded that it could be an X-type nitride, or the X-type ligand could be a nitride that does not exchange with dinitrogen. And again, this is under the idea that it was nitrogen to begin with. And so, to summarize where we're at this point, the structural data eliminated very early on the possibility of the X ligand being an oxide. Early on, the nitride was favored, despite being very similar in its deviance from the experimentally observed crystal. And I think the reason why it was favored is because the thermochemical data was strongly in the court of nitrogen, where a nitride ligand was expected to be much more exothermic in its binding to the cofactor, and the redox profile for a putative nitrogen ligand would be expected to also match the experimentally observed structure. So if you've noticed on this slide, I haven't included any spectroscopic data because Hoffman, 2 years later, repeated his same experiments where instead of measuring the entire enzyme, he then went ahead and isolated the FeMo cofactor and found that the Endor signals that he was attributing to the X-type ligand actually disappeared in the isolated enzyme. He further explored this by mutating the enzyme so that it couldn't turn over the catalytic cycle, perhaps rescinding his idea that the dinitrogen cycle reduction was actually responsible for this ligand, and found that even in a catalyst that was unable to turn over dinitrogen, the nitrogen signals disappeared. Instead, he pointed out a signal that he attributed to a carbon-13 nucleus and brought up the idea that it could be a carbide ligand. In response to this, though, Noodleman and co-workers performed a very similar experiment, came to the same conclusions, but they also probed the C13 signal a little bit more thoroughly and found that when they enriched the S-adenosylmethionine in their growth medium with C13, this signal didn't enrich at all, and they attributed instead to a sodium-23 nucleus that was dissolved in their solvent, in this case N-methylformamide. And so with that, we've gone ahead and ruled out a potential oxide on the basis of a structural incongruity. We've ruled out a carbide on the basis of electrochemistry and spectroscopy. And it appears that same spectroscopic data has ruled out a nitride. And so for a long time, it appeared that this study was back at square one, where it was still completely ambiguous what this X-type ligand was. And so this demanded a type of spectroscopy that did not require the long-range order that X-ray crystallography needed in order to observe a structure, but it also could not rely on the spin state of these nuclei because as early as 2003, Case and co-workers calculated that spin density on a bridging carbide and nitride and found that the expected spin density was actually very low. And I think Ian Dance put it best when he says that, ironically, the most interesting nuclei in this complex get almost none of the spin density, whereas the least interesting nuclei appear to get all of it. That being said, could a hyperfine EPR or Endor signal be observed even if an NMR active nucleus was in the center of this cofactor? And so, like I said, we needed a form of spectroscopy that didn't rely on these properties of the cofactor to provide structural evidence. And so this came in the form of XES spectroscopy. And for those not familiar with it, X-ray emission spectroscopy, or XES as I'll refer to it, effectively provides structural data without the need for long-range order. And the way this technique works is that you irradiate, in this case I'm using iron as an example, an iron nucleus with an X-ray of a given energy, and this excites a core inner shell electron up to an empty electronic state. This excited state then relaxes one of its 3D electrons down to fill this hole, and in doing so emits an X-ray of a characteristic energy that is directly related to the difference in energy between this valence shell and the core shell. And of course, the energy of this valence shell is directly related to the ligand frameworks around the nucleus. And so this technique has really been put to good use by Serena de Beer at Cornell, where she expected that this would provide a very high correlation between what ligands were bound to the metal center and what signal we get out of this study. And so what she does is she irradiates the complex and measures out its X-ray emission spectrum. And then she also goes through DFT studies to predict what these spectra should look like. And she finds that her predicted shifts for the energy of these emitted X-rays actually correlates very well with the experimentally observed shifts. although she's not able to very accurately predict the intensity of these bands, in recent years she's addressed this with a different DFT technique that's better able to predict those. But most importantly, she showed that different ligands around an iron nucleus were very easy to differentiate on the basis of that 3D to 1s transition. And she was able to identify a series of different light atom X ligands around an iron nucleus and found that the resolution between these bands was actually even the highest in the case of first row X ligands. So she went ahead and synthesized a number of structural model or synthetic models for these complexes. Here I've omitted the carbonyls that bridge the faces and edges of this model for clarity. And she made this type of structure with carbon, nitrogen, and oxygen. And when she measured out the emitted X-rays from here, found that the oxide, nitride, and carbide ligands could be very easily differentiated on the basis of X-ray emission spectroscopy. And so inspired by this, she teamed up with Marcus Ribby from UC Irvine, who had just recently published some work improving upon Shaw's isolation method, and found that when they performed the X-ray emission spectroscopy on the enzyme, or excuse me, the FeMo cofactor, they could identify this band down here that they predicted was responsible for the X-type ligand. So taking a look at the data, the experimental evidence was found to be in very high correlation with what one would predict for a carbide. And more importantly, the resolution between a carbide, nitride, and oxide ligand was exceedingly high. And so De Beer and Ribby came to the conclusion in this study that the bridging ligand was in fact a carbide. And I think this is the most compelling data I've presented so far as for the general identity of this ligand, and especially in terms of identifying it as a carbide. But you could ask whether this is conclusive or not on the basis of how off-path DFT studies got early researchers in this study. And so in the same issue of Science, for those still doubting this work, Rees returns with a 1 angstrom crystal structure, and by probing the electron density of that central X-type ligand compared to the predicted electron density for an oxygen, nitrogen, and carbon-based ligand, found that the experimentally observed electron density at the center of the cofactor correlated very well with a carbon, expected carbon and nucleus in that position. And one could argue here that, of course, Rees does not actually have 0.2 through 0.8 angstrom resolution data. And so a fair criticism is how reasonable are these data points, leaving only these three actually within his resolution. And even though the correlation is very high up there here between the three expected nuclei, I think together with De Beer's work, it paints a very nice picture of a carbide at the center of this cofactor. So to present what I've just told you on one slide, we show that the early work with computational carbide models actually very well or mimicked very well the predicted experimental bond lengths from the cofactor. And while magnetic spectroscopy was ambiguous, the X-ray emission spectroscopy predicted that the X ligand was a carbide with a very high probability on the basis of De Beer's previous work. And of course, the highest resolution crystal structure to date also came to the same conclusion, leading the field to currently rest on the idea that the central X ligand is indeed a carbide. So the question that is very fair for you to ask is, given that this is possibly the only carbide known in nature in an enzymatic active site, where does this arise from? And so to take a bit of a detour and look at some known discrete carbide complexes, as I showed you earlier, De Beer synthesized a cluster complex that has a central carbide ligand. And there are a couple of these known, and they were published originally by Churchill and coworkers in 1971. And these actually model very closely what we expect for FeMo Co, but there aren't very many of them in existence. Whether it's due to interest or just a general unavailability of them is something that I can't comment on at this time. But the other known carbides generally fall into the realm of either metathesis byproducts, where here we see that Grubb's catalyst can generate a ruthenium carbide during the olefin metathesis reaction. And in addition to that, a deprotonated alkylidine can also form a similar structure on molybdenum. Now, especially when we look at these two, these don't look very much like our FeMo cofactor, and that's largely because these are much more reactive than the carbide we see at the center of this enzyme. And in the case of these ruthenium carbides, they readily react to electrophiles to produce alkylidines. And if you're ever looking for a quick way to make a variant of Krebs catalyst, these are actually very useful for that. And of course, the deprotonated alkylidines are highly basic and will readily protonate back to the molybdenum alkylidine. And so the most common metal carbide known is actually the polymeric metal carbide. And you may remember tungsten carbide from the wedding bands that are becoming increasingly popular. And these metal carbides that are polymeric in nature are exceedingly hard and chemically inert. And this really doesn't sound like what we would characterize a biological living system as. And so the question could be posed is this carbide not only an outlier in terms of natural systems, but is it an outlier in terms of carbides as a whole? And so if we look at how it's formed, this FeMo-Co biosynthesis generally goes by the complexation of cysteine, iron, and S-adenosylmethionine to form this NIF-B protein, which then complexes with NIF-EN, which is itself a conglomerate of several other biomolecules, to form an ATP-activated cluster that subsequently produces FeMo-Co. And so Ribby and co-workers, in work that was published just actually about a month ago, identified S-adenosylmethionine as a potential source of this carbide ligand. And what they found was if they isolated, or they created a NIF-ENB fusion protein and incubated it with S-adenosylmethionine, they found that they formed FeMo-Co and S-adenosylhomocysteine along with 5-deoxyadenosine. And when they radiolabeled the methyl group of S-adenosylmethionine with either deuterium or C14, the deuterium was found to be incorporated into the 5-deoxyadenosine, one equivalent of it, of course, and that the C14 radiolabel was retained with very high conversion into the FeMo cofactor. And so on the basis of this, Ribby postulated that the methyl group first undergoes a radical abstraction, which results in this S-adenosyl deuteride. On the basis of the fact he doesn't observe the other two deuteriums, postulates that they're instead washed out to the solvent in two subsequent proton transfers, with his proposed intermediate being this ligated methylene radical that forms the cluster. And he doesn't have a lot of data to support this because those studies are still ongoing, but one could imagine presenting a radical trap to this enzyme on the basis of genetic modification using Peter Schultz's method, which is something I'd be happy to discuss after the talk. So the implications that this has for the enzyme as a whole is that we have an extremely rare, if not unique, carbide that is found in the center of the enzyme that is actually the only enzyme known to reduce dinitrogen to ammonia. Moreover, the mechanism by which this enzyme reduces dinitrogen is presently unknown, and likewise, what a carbide would do in a natural system is still also unknown, because this is the first time this has ever actually been observed. And as Norskov and co-workers pointed out early on, the incorporation of a carbide is energetically uphill. And the question to be asked is nature going so far out of its way to include this carbide in the center of this active site for reasons that go beyond its structural properties? And so I've reproduced the Haber-Bosch process down here to remind you how energy inefficient the laboratory version of this process is, and ask, if we better understand the active site of this enzyme, can we inform how we fix dinitrogen into ammonia for a more efficient Haber-Bosch process? So I put this slide in here specifically so that I can stop and say that the mechanism that this enzyme goes by is completely unknown. Much of the mechanistic work that has been published was ambiguous to begin with and relied on the idea that the central ligand was a carbide, a nitride. And since the discovery of this carbide or identification, no mechanistic studies have been published. And so what I'd like to do is try to assimilate some of the data that has been published over the years in the context of this carbide carbide and try to propose a mechanism that might take into account some of these features. So if we want to do this, the questions we need to answer are, what is the role of molybdenum versus the role of iron? So if we're talking about oddballs in this active site, of course, the carbide is the first one, but also the molybdenum is seemingly just hanging out in there. And so what is its role in fixing dinitrogen? Is the dinitrogen being ligated at molybdenum, or is it being ligated at iron? Moreover, how does dinitrogen bind in general? Is it ligated end-on, side-on in a polynuclear fashion or mononuclear fashion? And how does this impact the energetics of the reaction? And of course, now that we know it's a carbide, what would we expect this to have on the progress of the reaction? So early on, Dick Schrock, and actually for a number of years, he's been creating these structures that can produce ammonia from dinitrogen ligated complexes. And since the very start of this entire inquiry, he's proposed that the molybdenum is the business end of this enzyme. And he's shown that these molybdenum clusters that, or excuse me, molybdenum complexes that he synthesized bind dinitrogen, reduce it down to one equivalent of ammonia and a molybdenum nitride. This then reduces the nitride down to another equivalent of ammonia and binds another equivalent of dinitrogen, catalytically producing ammonia from dinitrogen. And so he's postulated that, again, the molybdenum is the most important metal in this complex. Now, of course, Jeremy Smith very recently showed that a iron 5 nitride, which is very similar in terms of its structure and electronics, can do the exact same thing. And I should point out that this complex here is just postulated. He identifies that the product of this reaction is iron 2, but doesn't actually identify its structure. But most importantly, we can see that while molybdenum nitrides can produce ammonia, so can iron nitrides. And so we could go ahead and look at these small molecule mimics as saying which metal is most likely to be involved in the reaction, which one is not. I can remind you that the Haber-Bosch process itself uses iron salts in its reaction. But I think the most compelling evidence for why molybdenum really isn't integral to this process comes in the figure I showed you at the very beginning, where if you starve the cell of molybdenum, it'll incorporate vanadium or iron, and the dinitrogen fixing ability of the enzyme is retained. I believe that the molybdenum is actually most intimately related in stabilizing the structure electronically. The key difference between the molybdenum and vanadium species being that the vanadium and iron-containing species are much more promiscuous with regard to reducing protons. So I'd like to propose that the iron nuclei around the X-type ligand are responsible for the binding of dinitrogen. So if we want to talk about the binding of dinitrogen, of course we want to look at how it is as a ligand, and we find that it's a very poor sigma donor and a weak pi acceptor, and it's really all around not a very good ligand. To date, all of the known small molecule dinitrogen complexes bind dinitrogen end-on, and it's found that polynuclear complexes reduce the barrier to subsequent complexation and formation, complexation of dinitrogen conversion to metal nitrides. And then these polynuclear clusters reduce those nitrides, in this case they're postulated, to ammonia with restoration of an iron 2 intermediate. And so on the basis of this, we have some relatively good data for how multiple iron centers can participate in binding and reducing dinitrogen. And Ian Dance, from very early on, and subsequently when the identity of a nitride was becoming a little more ambiguous, studied how dinitrogen can bind to these complexes. He identified a number of different ways it could bind, but found that the most favorable was one of these three, with this tetranuclear structure, or tetranuclear bound structure, being the most favorable mode of binding for the structure. And so on the basis of this, I'd like to propose that 4 iron atoms are involved in the binding of dinitrogen to form. There we go. Sorry. form an iron nitride. And of course, we could look at how the carbide is expected to interact in this setting. And we see that if we have a strong field ligand, we're expecting that the energy of the orbitals will increase, and this will lead to a preference for 2 electron pathways as opposed to 1 electron pathways. And this is what Smith observes in the case of his formation of an iron 5 nitride as opposed to an iron 3 nitride. And so on the basis of having that strong field ligand in there, I'd say that a polynuclear complex binds dinitrogen in a mu4 fashion, produces 2 iron 5 nitrides that are subsequently reduced down to 2 equivalents of ammonia, regenerating FeMo-Co, the resting state of the enzyme. And so, of course, if we wanted to probe this using small molecules, we could look at the work from the Betley group, in which they've synthesized these trinuclear iron species that are able to complex and digest azides. And while this is a relatively common process, this one is unique in its ability to do so at ambient temperatures without photolysis. And so this is an ability that's shared with nitrogenase, actually. If you feed nitrogenase azides, it will do just this. It'll extrude dinitrogen while forming metal nitrides. And so with this in mind, we could imagine similar complexes where we take a trinuclear cluster and synthesize what could be thought of as 1/2 of FeMo-Co. And we can tune this using actually what is pictured here as an alkylidine ligand, using some substituent on there. And if we take this a little bit further, could we show how these complexes would come together to form a similar intermediate, form the metal nitrides, and produce ammonia from those species? The key advantage here is that we'd be able to perform a kinetic analysis on this to show whether this is unimolecular or bimolecular in metal complex, and determine whether this binding motif is in fact operable. And regardless of what it tells us about the mechanism, is that if something like this does in fact occur, we could, it doesn't even have to be as efficient as nitrogenase. Even if it is half as efficient, we can produce ammonia in a much more energy efficient manner. And so with that, I'd like to go over what we've talked about so far. And the ultimate characterization of this complex with a bridging carbide was accomplished using XES spectroscopy and X-ray crystallography, where these provided evidence for the first time that this bridging X ligand was in fact a carbide. I went over the very recent work into the biosynthesis of this cluster, in which Marcus Ribby showed that the central carbide ligand comes from the methyl group of S-inosylmethionine, which is in and of itself a really interesting process wherein we produce quite a number of reductions onto this methyl group in an enzymatic system. And at the very end here, I took some time to describe to you how this can inform the mechanism of this enzyme and how that can lead itself into a more efficient Haber-Bosch process to produce ammonia for crop fertilization. And of course, this all comes back to the natural machinery that nature's provided us, with Azobacter vinlandi being the first to participate in this process. So with that, I'd like to thank the Chem 535 class led by Professor Paul Hergenrother, my advisor Allison Fout, and the entire Fout group for their support during this process, and of course your attention today, and I'd be happy to take any questions you may have.